In this video, we are going to discuss the process of transcription. Now let us see what is the central dogma. Now central dogma is basically conversion of DNA into mRNA and mRNA finally into protein. So information of DNA and mRNA is translated into protein. This process is called as what? Central dogma. Now central dogma, for the first part of it is transcription. So let us discuss the process of transcription. Now transcription is the process of conversion of information of DNA into mRNA by the action of enzyme DNA dependent RNA polymerase. So here the enzyme which is involved is DNA dependent RNA polymerase. So now this conversion takes place into 3 steps, but for those steps you require some unit. Okay, so let us see the requirements. Right, now the strand, it starts with DNA, right? So DNA has 2 strands. Okay, the one strand starts from 3' to 5', another from 5' to 3'. Now this, out of these 2 strands, 3' to 5' is a strand on which your RNA is going to be synthesized. So this strand is known as template strand. Now, this strand on which RNA synthesis does not take place, this strand is called as coding strand. So, the strand on which RNA synthesis takes place is called as template strand, and the strand on which RNA synthesis does not take place is called as coding strand. Now, template strand, it opens from 3' to 5' and coding from 5' to 3' as you can see here, right? Now the next term, so first requirement is you require template strand, okay, for the synthesis. Now second requirement is you require promoter. Okay, now what is promoter? Now, promoter is a sequence which is present upstream, that is near the 5' end of the coding strand. Okay, this is the sequence of DNA. Okay, now this sequence of DNA has TA sequence, therefore it is called as Tata box. So this sequence of DNA, that is promoter, it is present at 5' end, that is what? That is upstream. Upstream of what? Of the coding strand. Now the next requirement is the third requirement, that is, now here promoter is the sequence at which the process starts, okay, and terminator is the sequence, that is the next part, that is present at the 3' end of the coding strand. This sequence is the sequence at which the process stops. So next requirement is terminator. Terminator is the sequence which is present at what? Near the 3' end, okay, that is downstream of the coding strand. So let us see these requirements first. The first requirement, that is 3' to 5' is the template strand, 5' to 3' is the coding strand. Now template strand is a strand on which your mRNA is going to be synthesized. Now the process, the point at which this process of synthesis of mRNA starts is called as promoter. So this sequence is present around here, right, and terminator is present near the 3' and at which the process of synthesis of mRNA stops, right? So first template, coding strand, promoter and terminator. Now next after this is the sequences which are expressed in this mRNA, the sequences of DNA which are expressed in mRNA are called as exons, and the sequences which are not expressed in this mRNA are called as introns, right? So the DNA sequences which are expressed are called as exons and not expressed are called as introns. So we are done with the requirements. Now next after that is the steps in the process of transcription. Now let us see the steps here. Now what are the steps? There are 3 steps in the process of transcription. So let us see here. First you have 2 strands, that is 3' to 5'. This is your template strand, and 5' to 3', this is your coding strand. Now your promoter is the sequence around here, that is near the 5' end. So this is promoter sequence, and terminator is near the 3' end. So this is terminator, right? So at this promoter, at this point, the RNA polymerase binds. So this is the enzyme which is required here, that is RNA polymerase, which binds at what? At the promoter sequence. Okay, now this is the first step. So first step is what? Initiation. Initiation is the step in which what happens? The RNA polymerase binds. And what else is required here? You require initiation factor, that is sigma factor. Okay, so you require sigma unit, and another thing is RNA polymerase to start this process. As the process starts, now what happens here? There is separation of the DNA strand here, and there is formation of a bubble called as transcription bubble. Now this bubble is the point at which the process starts. So once the RNA polymerase starts moving from here, okay, from 3' into 5' end, the sigma factor, so here there is sigma factor, it will fall off once this polymerase starts moving. So first step is initiation, that is attachment of the polymerase, RNA polymerase to the promoter, okay, and formation of the bubble, and next after that is falling off of the sigma factor. Now as the RNA polymerase starts moving from 3' end to 5' end, the complementary bases are added. Now what happens is here your mRNA is going to be elongated. So second step is elongation. Now elongation is the process. Again here RNA polymerase acts, and what it does is adds the, so here there is addition of complementary bases. So complementary nucleotides are added here, and there is extension of RNA strand. So your RNA strand is going to elongate, so by the addition of complementary bases. Now what do you mean by this? For example, if in template DNA there is A, right? So in the your mRNA, that is new RNA which is going to form, here there will be U, right? For example, if here there is G in your DNA, then what will be there? What is the complementary base of GC, right? So in this way complementary bases are added, and there will be extension of the new RNA strand. Now thus your new RNA is formed. This RNA opens from, of course if here there is 3', this RNA opens from 5' to 3'. So new strand opens from what? 5' to 3'. Now what is the next thing? Now this process stops. Now where does it stop? At this point the process stops. Okay, for this you require rho factor. So rho factor helps in the stopping or the termination of this process, and the process stops. So last, that is third step is what? Termination. Termination means the point at which RNA strands stop synthesizing. The process of synthesis stops. Therefore, the last step is termination. This requires rho factor, Okay, and this is the point at which the RNA synthesis stops or terminates. Okay, so let us summarize this. The first step, initiation. This is the point at which RNA polymerase binds to the promoter sequence. Now this requires what? Sigma factor. So RNA polymerase and sigma factor are required. Now promoter is the sequence present near the 5' end, that is upstream. Now here the RNA synthesis starts. Once the RNA synthesis starts, the complementary bases are added and there will be elongation of this RNA strand. Now this elongation continues till what? Till the terminator. Now when this RNA synthesis, the RNA polymerase reaches here, right? This RNA polymerase reaches here, the process stops. Now for this termination, what is required? Terminator factor rho is required and the process stops. So in this way you have got the new RNA strand. So DNA converts into RNA by using what? By using DNA dependent RNA polymerase. That is it about the process. I hope you guys have understood. For more such videos, that is for next process, that is translation, please see my next videos. Thank you.